Eukaryotic cells rely on compartmentalization, but many critical reactions occur in organelles lacking lipid bilayers. These membraneless organelles, or biomolecular condensates, form via liquid-liquid phase separation. LLPS is a reversible physical process where macromolecules, primarily proteins and nucleic acids, demix from the surrounding cytoplasm or nucleoplasm, forming dense, liquid-like droplets. This process concentrates specific components, accelerating reaction kinetics by orders of magnitude while excluding others. The nucleolus, the site of ribosome biogenesis, is the most prominent example, functioning as a multi-layered liquid droplet within the nucleus. This dynamic organization allows the cell to rapidly reorganize its internal environment in response to external stimuli. The primary drivers of LLPS are weak multivalent interactions between scaffold molecules. These scaffolds are often intrinsically disordered proteins, or IDPs, which lack stable tertiary structure but contain multiple low-affinity binding motifs. These motifs include short linear interaction sequences, prion-Mike domains, and arginine-rich regions. Repetitive interactions between these motifs, mediated by pi-pi stacking, cation-pi interactions, and hydrophobic forces, create a dynamic polymer network. RNA molecules, acting as polyanionic scaffolds, also significantly tune condensate formation, often determining droplet size and stability by bridging multiple protein components. The concentration of these scaffold molecules must exceed a critical threshold for phase separation to initiate. Biomolecular condensates serve diverse regulatory roles essential for cellular homeostasis. Stress granules sequester messenger RNA and translation initiation factors during cellular stress, halting non-essential protein synthesis until conditions improve. Processing bodies, or P-bodies, are sites for messenger RNA decay and storage, regulating transcript stability and turnover. In the nucleus, Cajal bodies organize small nuclear ribonucleoprotein assembly, crucial for splicing. These structures are highly dynamic, rapidly dissolving or reforming in response to signaling pathways, allowing the cell to quickly adapt its metabolic state. The precise composition of these condensates dictate their specific biochemical function.
Initially, condensates exhibit liquid-like properties, characterized by rapid internal diffusion and fusion events, minimizing surface tension. However, over time, many condensates undergo a maturation process, transitioning into viscoelastic gels or even solid aggregates. This liquid-to-solid transition, often termed aging, involves increased cross-linking and reduced molecular mobility within the droplet. This change in material state can be functionally relevant, for example, stabilizing long-term memory storage in neurons, but it also underlies pathological aggregation. The transition kinetics are highly sensitive to temperature, pH, and post-translational modifications like phosphorylation, which alter the valency of the scaffold proteins. LLPS fundamentally organizes eukaryotic gene expression by concentrating transcriptional machinery at specific genomic loci. Superenhancers, large regulatory regions driving high levels of transcription, recruit high local concentrations of the mediator complex, RNA polymerase II, and specific transcription factors. This localized concentration forms transcriptional hubs, or condensates, which physically loop chromatin domains, facilitating efficient and coordinated gene activation. Disrupting the phase separation capacity of key transcription factors, such as the estrogen receptor, severely impairs target gene expression, demonstrating the necessity of the liquid environment for robust transcription. This mechanism ensures precise spatial and temporal control over the genome. Dysregulation of LLPS is strongly implicated in neurodegenerative diseases, including amyotrophic lateral sclerosis and frontotemporal dementia. Proteins like TDP43 and FUS, which normally participate in forming dynamic RNA protein condensates, contain prion-like domains that promote phase separation. Under pathological conditions, often triggered by mutations or cellular stress, these liquid droplets fail to dissolve and instead solidify into insoluble amyloid-like fibrils. This irreversible aggregation sequesters essential cellular components and leads to proteotoxicity, resulting in neuronal death. The transition from a functional liquid state to a toxic solid state represents a critical failure point in cellular quality control. Condensates represent a novel class of therapeutic targets, moving beyond traditional enzyme active sites. Modulating the material properties of pathological condensates offers a strategy to prevent aggregation. Small molecules can be designed to either stabilize the liquid state, preventing solidification, or promote dissolution by disrupting weak multivalent interactions. For example, targeting the interaction motifs of FUS or TDP43 could prevent their pathological transition. Furthermore, high-throughput screening assays are being developed to identify compounds that shift the critical concentration required for phase separation, effectively dissolving unwanted aggregates or stabilizing functional ones.
LLPS is not exclusive to animal cells. In plants, stress responses frequently involve the rapid formation of cytoplasmic condensates containing heat shock proteins and specific messenger RNAs, protecting the translational machinery from damage during heat or drought. Furthermore, prokaryotes utilize analogous mechanisms. Bacterial microcompartments, while sometimes encased by protein shells, rely on phase separation principles to concentrate metabolic enzymes, such as those involved in carbon fixation or propanediol utilization, enhancing efficiency by minimizing diffusion distance. This suggests LLPS is a universal, evolutionarily conserved strategy for cellular organization across all domains of life, optimizing biochemical throughput. Understanding the precise molecular grammar governing LLPS allows for rational design of synthetic systems. Researchers are engineering synthetic condensates using orthogonal protein domains to recruit specific enzymes, creating programmable reaction centers that function independently of native cellular processes. This technology promises breakthroughs in biomanufacturing, allowing precise control over reaction yields and product purity by spatially confining reaction intermediates. For instance, creating artificial metabolic pathways within designer condensates minimizes toxic side product accumulation. The future of cellular engineering hinges on mastering this fundamental physical principle, enabling the construction of highly efficient, customized cellular factories.